I'd like to thank you for joining this morning's webinar, which is the second webinar of 6 in a series by the IQ 3R's Reproducibility Working Group. This webinar is sponsored by the IQ 3R's Translational and Predictive Sciences Leadership Group and the Statistics Leadership Group. Today's webinar is entitled Replicability and Reproducibility in Animal Studies, and it is presented by Stephen Novick of Eli Lilly and moderated by Xin Huang from AbbVie.
Steve Novick is a 20-plus year seasoned biostatistician within the pharmaceutical industry. He is currently executive director and leads the Discovery Statistics Group at Eli Lilly. Steve's publication history includes articles and book chapters, primarily on the CMC and preclinical topics. So Steve is an ASA fellow and serves on the executive committee.
Trials have failed for efficacy, safety, or both. Well, what does that mean? If you fail for efficacy, that means that our animal studies efficacy either didn't translate properly or we weren't really sure. We weren't as confident as we could be, or some other error. If it failed in safety in a clinical trial, that could also be some sort of translatability problem from animals to humans, but it could also be that we just weren't sure that the results would repeat properly. The second bullet point says only 18% of drugs were successful in phase 2. Well, phase 2 is for sure efficacy. And so again, that either means that we didn't have good medicines translation, which can happen, or we weren't sure that the study we ran in animals would have repeated. And so that's what we're going to talk about today, is repeatability and also reproducibility. Next. So we often operate under what are called the 3 R's. The 3 R's are replace, refine, and reduce. We're always trying to lower the number of animals per study, which I guess is reduce, and replace. It tries to drive some innovation. So we always have this in the background of animal studies, where we're trying to create better analyses, better use of our data, better modeling approaches. Next one. And we also operate under the ARRIVE guidelines. ARRIVE stands for Animal Research Reporting of in vivo experiments, and it's something that you'd probably like to Google if you don't know the ARRIVE guidelines. Next, these are part of our good statistical practice. When we apply good statistical practice, which is something else you can Google and find out about. Essentially, this is what we do as statisticians. We ensure that studies have the right experimental design, that we've got controls, that we have the right data analysis in place, we've powered the study properly, and so on. And when we do this, we hope that our study results are both meaningful and interpretable. Next. So the question that I posed to myself, I guess, about 5 years ago is, are the ARRIVE guidelines good enough? Can we be confident from the results of a single study? Next one. So supposing we conducted a study with the ARRIVE guidelines, this data is totally made up, but that doesn't matter. And we have here treatments A and B, and the statistical test, we'd like to show that B is bigger than A. We'd like to show that the whatever the assay response is. This assay response is supposed to be in vivo, some sort of animal study. I've got 8 data points for A and 8 data points for B, so 8 animals in each group. I've got the delta there is supposed to be the truth. So delta is 14. And I ran a t-test on this data, and I found that the p-value was less than 0.0001. So almost everybody would say, I'm really certain that I'm going to go with the alternative H1, that the mean of B is bigger than A. Next. So some people would call this significant, as I put that word in there just to generate some controversy. You may know that the ASA has a paper by Wasserman, and we're trying to get away from the word significant, and there's a whole series of papers on making statements like, there's a strong evidence that B is bigger than A. But in any case, we're going to conclude that B is bigger than A. And if this was the animal phase for efficacy, we would move the drug asset to the next phase. We would perform safety, or if we already perform safety, maybe we move to the next phase where we make a decision about moving to a phase 1 human trial. Next. But I want you to understand that this single study, this p-value, it doesn't matter as a p-value. We could have used a Bayesian probability. We can only conclude that B is bigger than A this time. In this study, there's no repeatability here. We don't know if we had reperformed this study, would we have seen a mean difference of that magnitude and drawn the same conclusion that B was bigger than A. Next slide. So here's a riddle. My kids are adults now, But back when they were small, they would tell me this riddle. Pete and Repeat went fishing, and Pete fell off the boat. Who was left on the boat? And next. The answer is Repeat, and then they would say the riddle again. That was their joke. But this was my fun way of defining repeat. So a repeat is a reperformance of the study in whole. It doesn't have to be exactly the same conditions, but the conditions should be at least similar. Probably conducted at a different time point, probably or possibly with different investigators, possibly in a different laboratory. Next. So there's a couple of papers that if you have these slides, you'll see in the notes the references that I placed there. It was found about a decade ago that repeatability of preclinical experiments was as low as 10%. And that is a problem. I think the latest paper I saw, repeatability was up to 50%, which is a whole lot better, but still not fantastic. Next. So I want to propose a kind of a mathematical statistical solution, a way of modeling our data. But first I want you to think about two kinds of random error. If I repeated the study many times, I would certainly have study error. I would have study error, and I would have within study error. And so we could think of that like it's a main effect error. But we could also have study by treatment error, which you could think of as a random interaction. Next. So first, if we only think about study error, we might fit a model where we have a response variable y, which depends on the treatment and is also that J indexes the study, where we would have a mean that's treatment A or treatment B, and we would have study. So study could be study 1, study 2, study 3, like that, and that would have random error, and then we would have the subject error, whatever the animal error within the study is, as that last error piece. Next. If I want to compare means A and B and include the study term, I would have mean of B plus study J, whatever J is. J is, it could be J prime, the next study, and then subtract off mean of A plus study. It could also be J prime for the next study. And the study terms cancel out, and I'm just left with mean of B minus mean of A. Next. Which means that I don't really need to think about the study error. I only have to model study error. And if I had many studies in a row, if I had repeated the study, I'm expecting with a model like this that I'll get the same mu of B minus mu of A and reach the same decision. So next one will show you next slide shows a picture. Next slide, yeah. So here, if we only have study error, the main effect error, on the left-hand side, we had the original study with delta of 14. And if I repeat it, even though all the data will move up and down together, the important word there is it moves together. And delta is still 14. And if I repeat it a second time, repeat number 2, I still get a delta 14. So there's no need to repeat here. If all I have is the main effect of study error, then I have absolutely no need to repeat the study because I'm expecting to get the same answer, make the same decision. Next one. But if I have study by treatment error, next, then when I do the mean comparison, including the study errors, I think if you go next, it's going to do some crossouts. The study terms cancel out, but I am left with the study by treatment terms. And so any inference I make that includes the study error will retain the study by treatment terms. And This means that I would need to repeat my studies in order to estimate those extra terms. Next, please. Next. Graphically, this means that I'm expecting, I like to think of it like an accordion. Sometimes it's here, sometimes it squeezes together, and sometimes it pulls back out. So when I repeated number 1, the truth was that the means grew closer together. And when I repeated number 2, the truth was that the means moved further apart. And so just from my original study, that wasn't enough for me to be absolutely certain that if I repeated the study, I should reach the same conclusion. Next one. So this was a little problem when we went to publish, because we originally used the word reproducible, and there seemed to be some problems in the literature. So we went with Patil. Patil says the result is replicable if you can consistently reach the same results after reperforming the experiment. Mohan called the same thing reproducible, but we're not going to go with the Mohan definition. So next, we're just going to toss away the Mohan definition. Next. All right. So what are some barriers to replicability? Well, one of those barriers is if I don't use good statistical practice, if I don't use blinding, or if I don't randomize the animals into treatment groups. There's so many things that can go wrong, but let's assume that we are using good statistical practice. Secondly, is maybe your study has a lack of good quality control. Lack of good quality, where maybe if you looked at your control animals, you would see that there's a problem with the quality of the study. So did you interpret your study results even though the study itself did not have acceptable quality? Third one is p-value hacking. P-value hacking occurs when a person might say, I really wish I had a p-value less than 0.05, and I will have a p-value less than 0.05 if I just remove this one animal from the study. And so the scientist might say, this one animal is causing me some concern. The value is a little bit out of trend. I'm going to remove it. And suddenly the p-value is 0.04. And that's why I wrote the correct p-value with italics to indicate that I'm being a little sarcastic. It's not the correct p-value. Next. And of course, it could be that the study by treatment random effect is quite large relative to the mean difference that you're looking for. And so had you repeated, if you did everything else right, it's possible that your study by treatment random effect is large enough that if you repeated, you would reach a different conclusion. Next. So let's assume that you do everything correct, but you really don't want to spend money on repeating an in vivo study. So there are some solutions in the literature, including my own. And first, I'll list some competitor methods. And it's not that I need for you to choose one or the other, mine versus theirs. They're all good methods. So Focal in 2018 thought about breaking up an experiment across different labs. So if you had 2 or 3 laboratories and you had 60 animals, you might do 20 animals in each laboratory. And then when you combine the results together, you'll get some idea of study by laboratory by treatment interaction. Natasha Karp gave a talk just last week to the IQ Consortium in this series. And she came up with a nice method called the multi-batch design, where instead of breaking up the experiment across laboratories, you break it up within a laboratory, but across time. So maybe you've got a 20-week study, and you decide to start 20 animals at week 0, and then 20 more animals 1 week later, and 20 animals 1 week later. So instead of being a 20-week study, it's a 22-week study. You just have to be a bit careful about bookkeeping, but this is a way to generate some within lab, but I guess you would call it, she calls it multi-batch. So you would have batch by treatment interaction. Next. So the method that I propose with my co-author doesn't require any physical repeating. You still would have to have your primary study. Next. And so here it is, your original study. You've got a certain number of animals, and you know, the problem is the scientist doesn't want to break up the study, doesn't want to take the extra time, doesn't have the extra laboratory. And when they do, the batch method or the focal method is great, and I would be a big advocate for that. But if you just have your one laboratory and you're running out of time, then what you have is in the middle bullet points as our proposal is you're looking for the predicted mean difference for a future study. And so that would be that mu B minus mu A plus the difference of the study by treatment random terms. That's where we use this J prime to indicate the future study. If this is a true statement, if you can declare this as your alternative hypothesis, then your result can be declared replicable. Next. Of course, with just this one data set, I don't have a study by treatment error term. So that's a problem. Next one. So, okay, you know, mea culpa, we can't estimate study by treatment, but maybe we have some historical data that has enough information that will allow me to estimate study by treatment, even if the treatments are not treatments A and B. So one example that a few scientists indicated to us is a lot of in vivo studies have vehicle control groups with a plus vehicle and a control group without the vehicle. That would be the minus. So plus is the yes vehicle control group, and minus is a negative control group without vehicle. Next one. And a graph might look like we might have 5 studies with plus and minus vehicle control. You might have a vehicle control and a comparator control in each study. You don't have to have the same pairings in each study, but you have to have pairings of something across different studies in order to estimate study by treatment interaction. And so here you could see that the mean differences are not, well, these are just the point estimates, but they're not the same from study to study. Next. In this case, we get a study by treatment standard deviation estimate of 1.5, and we can use that information somehow. Next one. In our paper, the 2021 paper, we show you how to use these calculations with an ancient method called method of moments, or the more frequently used one, the Kenward-Roger method, which is available in proc mixed and also in R using LMER and the EM means libraries. And of course, if you're a modern statistical learner using something developed in late 1800s, you can be a Bayesian. Next one. So Bayesians would just call this the posterior predictive distribution, but for the sake of this webinar, we're going to show the Kenward-Roger method. Go ahead. Next. Next. So Kenward-Roger, you would put all of your data together. So if I'm in SAS using proc mixed, I'm going to put together my 2 data sets, my historical data and my original data. Or if I'm using LMER, I do the same thing. And so I would have a main effect for study and an interaction term for study by treatment as random. Those would be in the random statement. And DDFM equal KR, or I think there's a KR2 now. Next. Now, when I compare mean of B minus mean of A, I get a p-value 0.005. Remember, the original p-value was less than 0.0001, and now it's 0.005, which is quite a deal bigger, but still small enough that I can now say that B is bigger than A. And if I had repeated this study, I would still say that B is bigger than A. So I am so much more certain that B is bigger than A than I was when I only had the original study data on its own and made the statement that B was bigger than A this time. Okay, next one. And if you go look at that paper, it also shows how to calculate sample size and statistical power when you want to show replicability. Right, next one. All right, so in summary, if you think about replicability and incorporate that into your data analysis plan, you should end up with higher confidence that the results from the animal will be replicable in humans, as long as the animal model translates well to humans. This method doesn't require additional resources, neither does the CARP or the, I forget the name right now, but the other method. It doesn't require many more resources in any case, whether you split up the experiment within a lab or you go and look for historical data. And by the way, you could do all three methods if you really want to use your historical data, break up your experiment within a lab and across labs. There's nothing to prevent you from doing that. Next one. So that's my take on replicability. Now for reproducibility. So reproducibility, you could think that kind of an old statistical programming paradigm was to have somebody look over your shoulder as you wrote a program to make sure you didn't make an error. So you want to avoid all sorts of mistakes, whether it's writing your script or your point and click software and writing your report. So reproducibility has to do with reproducing the statistical analysis and report writing. Next one. This is definitely less sexy. When you think about modeling data, you can think about, oh, how am I going to reuse my historical data? How am I going to make this efficient design? Reproducibility is more about bookkeeping and being neat about it. Next slide. So going back to Patil, he says that a result is reproducible if I use the same data, the same analysis plan, and potentially even the same computer code. I should obtain the same results obtained by a different data analyst, a different data scientist. And there's all sorts of errors that can come into play, whether they're in data management, control, code control, and reporting. Next one. These are some older references. I suppose they're older by now, but they are as true then as they are today. I suppose you pronounce that author's name, Ioannidis, in 2009. They found in microarray data that they couldn't produce the same result from the raw data. Well, maybe that's because, just like in the Baggerly example with poor documentation, they didn't know possibly how to normalize the data. Maybe data were normalized differently each time, and then the data analysis plan just couldn't be followed because the documentation wasn't there. In the Nekrutenko and Taylor I've experienced this one myself with not with next generation sequence, but with other data. They claimed that in 26 out of 50 data analyses, they couldn't get access to the original data or the original script, like an R script or a SAS script, or the original version of the software it ran on. If you've run, I don't know if you're an R user like me, but when you use libraries, they're not always backwards compatible. Some of my ggplot code that I wrote 10 or 15 years ago doesn't function anymore because the commands were deprecated. And so if I don't have the original R code, the software itself, the executable, it won't run. And so all three of those are problems. Next. So I'm going to go through these 6 bullet points, and it won't take me too long. I'll leave time for questions. Having a consistent way to organize your projects. We're talking about file management. Secondly, good data management. Thirdly, performing your analysis using a scripted language, a scripting language. Version control for your scripts, your computer code. Using a consistent coding style. And then finally, avoiding copy and paste errors in your report writing. Next one. Yes, it's a little boring, but I've run into all of these problems and wish that I had followed my advice from when we wrote this book chapter. Next one. So how can I organize my folder system and manage my data? Well, this is the very simplest way we could think of is you have a consistent folder style where project name might be the top level. And then the next level would have folders called analysis, data, documents, and reports. The analysis would contain your computer code. The data would contain your current data and a folder below it, a subfolder called archive. So if you're like me, you've experienced this many times where you have a data set, you've run your analysis on the data set, and the scientist or engineer will say 2 or 3 weeks later, oh, I made a mistake. Here's the new data set. But you don't want to throw away the old one. And so you put the old one in the archive and you put your current data in your data folder. Next, please. It's important that the data, your most recent data goes here. So there's no confusion about which is the data set you analyzed. So if somebody looked at this folder 5 years from now, they will know that is the data set. And please make it read-only so that no changes can be made over the course of those 5 years. The documents folder will contain some instructions, maybe study protocol, whatever clarifications might come in some emails, maybe slide decks that explain what testing needs to be done. And then reports folder would have the output of the analysis. Next one. Using a scripting language. Well, a lot of our point and click software systems don't leave an audit trail. Or if they do have an audit trail, it's not always clear what the order of the steps were. But if you use a scripting language like R, Python, SAS, or maybe you like to use Java, C#, whatever programming language you use, not all, but most of our programming languages are linear in that they execute instructions from top to bottom and in a very easy to understand first, second, third, fourth, fifth manner. And so somebody can read your code and know what the order of operations was in your computer program. These also permit commenting. Imagine if you used point and click software without an audit trail, you would have to write a document to let people know, I pointed and clicked here, I pointed and clicked there, and this is why I did it, and so on. Scripting languages also allow for debugging, which can be important, but I suppose you would have all the debugging finished by the time you finalize your code. And finally, you can track your analysis as you go through output logs and results, which is something that you can save so that 5 years from now, if somebody needed to reproduce your analysis, they could check your output logs and your results and see that they got the same values. Next one. Version control. So a lot of departments are moving towards software versioning repository like GitHub, and so even though you're keeping your code in one folder, you might have a copy of that code in GitHub, which helps you with code changes. So for example, if you ran an analysis and the scientist asked you for more, and you can add those pieces of code, and you would have just a new version of the same document. So you wouldn't have to create document after document, and everybody would know what's the most recent version of your code. And you could also put in textual justifications for why you made those changes. Now, supposing you wrote a function and you decided you're going to make big changes to that function, and you run the code and the new function doesn't work, but you saved your new file over the old one, so the old one is gone. Well, with version control, it's not gone. You could just revert back to an earlier version, the version that worked, and you could start again. So this idea of branching off and trying things in your own sandbox and then merging the code back, for certain projects, that this will make sense. Next one. Okay, this one I actually caught myself on just the other day. I was looking at a piece of code that I wrote about 10 years ago, and I couldn't understand what I did. So that third bullet point is think about coding for your future self. Having a consistent coding style will not only help a team that you're working on, a team of programmers, to read each other's code, or the person who's trying to reproduce your code to help them understand what it is, but even for your future self to know that you have a consistent style. There are some style guides like Tidyverse and Google computer coding styles, which are specific for R or Python, Java, and SAS. So you could look those up and see what those are, and then everybody agrees, okay, this is generally how we'll write our computer code. Next one. And I think this is the last one, automated report writing. This was especially important when I was working in manufacturing, that oftentimes we would have 10 tables and 10 graphs that would go into a document. And the first time I would copy and paste all 10 graphs and all 10 tables into a Word document and annotate all those tables with legends and text around it. And then I would get, the scientist would call and say, oh, I made an error in the data. Here's the new data set. And I would have to do the whole thing again. Now, not only could I have made copy and paste errors, but I had to do the whole thing again, meaning that I had to copy the tables. And sometimes I did make an error copying the wrong graph or the wrong table in the wrong place. The use of markdown languages like R Markdown, Sweave. There's another completely different paradigm called Office R, which allows you to create Word and PowerPoint documents. helps you to avoid copy and paste errors and keep things in the correct order. Next one. So all of these things will help you with replicability. Replicability will provide you with more confidence that your study results, had you reperform the experiment, then you would reach the same conclusion. And that should mean that we're more confident by the time we get to a clinical trial. And reproducible, meaning that the code you've written, the report that you've written, that you've got all the pieces correct, and another person could have taken your code and created the same exact report. Imagine if another person took your code and couldn't reproduce your report. What would that mean? It would mean that it's probably copy and paste error. Maybe there was a coding error that you missed, or maybe different versions of R or different versions of SAS gave slightly different answers. And how does one interpret that? So again, just gives you more confidence that the results you see, if you're applying this to animal studies, will translate better to the clinic. And so that's what I have today. Next one. So I thank you all for attending and listening, and I hope that was worthwhile.
Thank you, Steve. So if in the audience we have any question, please enter it into the Q&A box. And while we are waiting for questions, maybe I have some questions for you, Steve. The first one is, have you applied any of the replicability methods in your work, and how was it received by the scientific team? How things changed after your implementation?
So I've got a lot of Bayesian training, and so it's natural for me to want to reach into the historical data and see if we can apply that. And there's really two. I like to think there's two ways we can apply historical information. One method is to gain more confidence for this study. So not to think about study by study interactions. And the second way is to look at the posterior predictive distribution. And so I had a few metabolic studies where we took a look at we calculated it both ways, to be honest, because the scientist wasn't willing to bet the farm on replicability p-value or replicability posterior predictive distribution, and so we calculated probabilities both ways using historical data, and generally it just gave us a lot more confidence in the results that we were able to say. In both cases that we used it, we found that the probability was high enough to show replicability, and so that gave us more confidence in our results.
Okay, thank you. A question from the audience is, do you have any advice on how to handle data outliers in an objective way to avoid the p-value hacking? Well,
an outlier has nothing to do with p-value hacking. I love that question. So I like to think of there's you could bin observations into this observation looks like a typical observation and it's acceptable. The observation is completely unacceptable. Wow, based on my experience, this is an outlier. Something must have gone wrong. And you could either do a root cause investigation to prove why it's an outlier, or you could just you could with less confidence say that I've never seen a value like this before. It's certainly an outlier. And then you have the one that's in between. That's where the p-value hacking comes into play. You know, if you're expecting data to fall between 8 to 12, and you get a value of 55, you know that's really far away, and that data point should be removed. But if you get a value, it should be between 8 and 12, you get a value 14, and removing that value will allow the p-value to go less than 0.05. Well, that's p-value hacking. That's dangerous and should be avoided.
See, thank you. Another question from the audience is if there are some other factors that you didn't collect, but maybe it's important for the reproducibility, then how do you handle those unknown factors?
So putting the question a different way, if I reproduced, if I replicated the study, if I reperformed the study, and I didn't get reach a consistent conclusion, what would the problem be? And what if it's a problem that I hadn't considered? And who was it? Donald Rumsfeld said there were unknown unknowns. This is called Simpson's paradox, right? We don't know what's going to cause what's missing from the study, and so if you've ever created an Ishikawa diagram, or what's also called a fishbone diagram, you could think of all the inputs that could affect your outcome. And typically we don't run a study that has all of those different inputs. We just select the ones we feel are important. But if we have an entire list of inputs and we think we may have missed one, we can go back into it and see. Hopefully we made some measurements to see if anything that we missed from our list was important. But if we don't know what it is, then we still don't know, and we just have an inconclusive decision.
Yeah, that's true. So these comments from me, what I observe in the preclinical safety, we have some regulatory guidance regarding use of the vehicle control data in the historical database, and then to establish a process control chart so that you'll be able to evaluate whether there is some drifting or some change in your historical vehicle data, and then you can go back to check what are the factors that has making this switch, these changes. So this is related to your fishbone chart to identify what are the potential factors that we didn't consider, but potentially can be important and contributing to the reproducibility.
And by the way, that's a great comment. Monitoring is part of the ARRIVE guidelines, and so you should be looking at your. It sounds like manufacturing, but you should be looking at control charts and look for drifting and quality of data that might be lower than expected.
Yeah. So it seems that lots of statistics effort involved. Not only the patients, but also you are building the historical data and also use this process control charts. So another question from the audience is that there are so many statistics involved, and how do you suggest for a non-statistician to use all of this when they don't have a formal training in statistics?
I'm not sure where to go with that, only because there's so many possibilities. We use statistics every day, whether you're a statistician or not, to make a decision. You use whatever historical information you have in your brain to kind of calculate probability of taking the right action. Should I buy this new car? Should I eat eggs for breakfast today? Whatever it is, maybe unbeknownst to you, you're using some statistics in there. But to apply statistics to study data, well, firstly, you could just partner with a statistician. So if you don't have a statistician, you might find one at a university who's willing to partner with you, even if that's a graduate student. If you're fortunate enough to work at a big company like I do, Eli Lilly, then we have a whole group of 15 preclinical statisticians, and we have a few more in other locations. And so you would partner with us. We offer training and guidance that are quite pointed towards the types of studies that you might embark upon. And there are statistical consultants out there who, for a fee, will provide you with training. You can attend statistics conferences. You can read statistical, some of the simpler statistical literature. If you typed into a search engine control charts, there's probably 20 very easy to follow articles in there about how to create control charts, even in Excel. So learning statistical philosophy, that's hard. That requires a lot of either school or study, and then living it. It's like a religion, right? You live your religion, and that helps you to get a better understanding of your religion. If you live your philosophy, helps you get a better understanding of your philosophy. And short on that, you could just learn the tools and apply the tools, knowing that it's the quote unquote right thing to do. I hope that answered the question.
Yeah, that's great. I think this aligns with all these are related to the scientific philosophy, right? How to conduct a robust science. Statistics certainly is a great tool to achieve the scientific exploration. Another question I might have is the automation. Right? How do you see if we make all this into automation, will it be a great approach to ensure the reproducibility and also replicability?
I'm really excited.
Yeah,
I'm really excited by that question. My former colleagues at AstraZeneca were using a tool called Knowledge Graph, but I think a newer tool that people are going to use, it will be ChatGPT or similar. And you could picture a day, maybe not in the far future, where ChatGPT oversees a large database and understands that the study I'm about to conduct is related through the metadata that we store, related to a series of studies that we have in our internal database, but also might scour the literature and pull up a few relevant papers and incorporate the data from those papers to create essentially that study by treatment interaction term, or whatever kind of study by treatment term we need to draw inference. So I think in terms of replicability, we'll be able to draw on historical data in the future. That should be easy. Reproducibility, as long as we use a tool like R Markdown, reproducibility will be pretty automatic. And not to emphasize ChatGPT too much. By the way, I've used ChatGPT and have had some pretty catastrophic failures with it, so I don't think it's quite ready. But ChatGPT might be able to write your R Markdown code in the future. You just tell it what you want, and it could, based on your previous scripts, figure out what your R Markdown document will look like, and then you just have to read it. Of course, it'll be reproducible if you have the same R executable program in libraries that you used when you created the document in the first place.
Yeah, certainly we are looking to the future, right? It's a rapid changing field, and in a few years, a lot of these advancements can help us to tackle these challenges. Yep. We have another question from the audience. It's related to the genetic drifting. It seems, have you found genetic drift in knockout controls, like generations from initial breeding pairs, to be an issue with your method? I found that in many instances, it's difficult to even obtain the data. I found that this genetic drift in the knockout mice many times results in the loss of phenotype at times. Yeah.
I don't have experience there.
Mm.
I can only offer the advice that you could try to incorporate the drift into your analysis to use the historical data. That's what Bayesians do. Right? Bayesians say, this is my historical information, but something has changed about it, and so I'm less certain, or I have to put it through some sort of function to kind of modernize my historical data. But aside from that, I haven't worked with replicability in that system.
Okay, thank you. So we are on top of the hour. Thank you for all the audience for joining us, and thank you, Steve, for giving us this very insightful presentation.
And thank you again for the invitation.
Okay.
Hope everybody has a great day.
Thanks, everyone. Have a great day.